Our last speaker before lunch is Dr. Nathaniel Hogrebe, who's a senior postdoc in Jeff Millman's lab. Jeff couldn't be here and highly recommended Dr. Hogrebe. I hope more PIs are willing to do that for their people. So thank you, Jeff. Dr. Hogrebe is the first author on a Nature Biotech paper and a Nature Protocols paper on how to differentiate pancreatic beta cells. This is the person to call if you are having trouble with your beta cell differentiation. Hear that, Vijay? So his title is Stem Cell Derived Islets for the Treatment of Diabetes.
Thank you very much. And if you ask me questions, I'll first tell you to read the protocols paper. That's what I do to everybody in my lab. So, alrighty. Okay, so today I'll first just kind of be giving an overview of how we can, how stem cell derived islets can be used for treating diabetes as a cell therapy, and then discussing our newest methodology for making these beta cells. And hopefully in the process, I can demonstrate how culture format can influence your differentiation success. And then finally, our efforts to scale up this process to generate billions of cells per batch. So first, a little bit about the pancreas. So most of the pancreas is made up of exocrine tissue that secretes digestive enzymes into the upper portion of the small intestine. And then scattered throughout the rest of the pancreas are these clusters of endocrine cells called the islets of Langerhans. And the two most abundant cell types are the alpha cells and the beta cells. And so your alpha cells will secrete glucagon, and among other things, it tells your liver to release some of its glucose stores, and this raises your blood sugar during periods of fasting. And then your beta cells secrete insulin, which allows the sugar in your blood to enter cells throughout the body so that they can use it as for energy. And so when your beta cells can sense changes in your blood glucose levels, such as when you eat, and then secrete the appropriate amount of insulin in response. And so in type 1 diabetes, there's an autoimmune attack that selectively destroys the beta cells. So these patients don't secrete any insulin, so they have to take insulin injections every day in order to survive. Type 2 diabetes is a little more heterogeneous disease. There's some sort of defect in how insulin is secreted or processed, but ultimately a lot of these patients do end up have to go on insulin as the disease progresses. Replacing these lost beta cells can potentially provide a functional cure for diabetes. And this concept has been shown to work by transplanting cadaveric human islets into patients. But there are two main obstacles to making this therapy widespread use. So one, we need to generate an unlimited supply of beta cells that can sense changes in blood glucose levels and then secrete the appropriate amount of insulin. And then also we have to deal with the problem of immune rejection, because transplanting these cells is pretty much like any other transplanted organ. But today I'll be focusing mainly on the generation of these cells, but we do have work going on that is dealing with the immune rejection problem. So the first generation of protocols were published in 2014 by a few different groups, and my PI was one of the co-first authors on the cell paper. And this work spun out a company called Sema Therapeutics that continued to work on this technology, and then a few years ago was bought by the pharmaceutical company Vertex. And last year around this time, they transplanted the first patients with these cells. Since these were the first patients, they only gave about half of their target dose, and these patients do have to be on immunosuppressants. But in their latest update over the summer, they reported that patient 1 is completely off insulin now, and his blood sugar is in range about 100% of the time. Patient 2 was a little less successful, but they still had a 30% reduction in insulin requirements. and their blood sugars in range about 50% of the time. So this first clinical trial showing that these cells are safe to use and also that they work at controlling blood glucose levels, but there's still some variability. And so there's definitely room to improve the cells. And so a major goal research area in our lab is to make these cells better so that we can improve clinical outcomes. So for example, if your beta cells secrete twice as much insulin, you would need to transplant half as many islets. And so from a clinical perspective, this is fairly significant in terms of both the volume of graft that you have to transplant and also production costs, because these cells are quite expensive to make. And so now I'm going to discuss the improvements we've made to these cells over the past couple of years and the development of the current version of the protocol that we use today. And so there's a number of different versions of these protocols out there for making beta cells, but all of them generally follow the same strategy where we try to mimic embryonic development by turning on and off specific signaling pathways with the timed application of growth factors and small molecules, and driving them through these several intermediate cell types on their way to becoming beta cells. And so the original protocol was done in suspension culture in spinner flasks like this one to propagate the stem cells as clusters and then differentiate them. And so when I joined the lab a number of years ago, one of the first things that we did was create a new maturation media at the last stage of the protocol, which significantly increased the performance of these cells. So here we have a glucose stimulated insulin secretion assay where we incubate the cells at low glucose and high glucose for an hour each, and then measure the amount of insulin that they secrete. So in the middle, you can see the insulin secretion of the original 2014 protocol. And on the left, you can see the increased amount of insulin that these cells secrete with this new maturation media. And so we can also measure insulin secretion every few minutes to get a greater resolution on the kinetics of insulin release. So primary human islets have this characteristic burst of insulin secretion upon stimulation with glucose, which we call first phase. And then after about 10 minutes, insulin secretion continues, but at a reduced level during the second phase. And then importantly, secretion is able to shut off when the cells are returned to low glucose. And so you can see here that the cells from the 2014 protocol had very low insulin secretion in this assay, whereas in our new maturation media, they gained a dynamic insulin secretion profile similar to that of human islets, although at slightly lower levels. And you can see the acquisition of their ability to secrete insulin over time. So at 5 days into this stage 6, they aren't secreting much, but then their function, they gain the ability to secrete insulin, and their function peaks between 2 and 4 weeks into this stage. And so the next advancement in the lab and what I've been working on the past couple of years was the development of a new hybrid version of the protocol that we now use in the lab today, where we actually specify the beta cells in planar culture first and then aggregate the cells at the end. And I know that there's often a push to always move towards suspension culture, but that may not always be necessarily the best for your particular differentiation. And so in our case, we found that the system actually generates cells that are much more functional, and we have the advantage of differentiating cells from a much wider range of cell lines. And so I'm going to first talk about the workflow of generating these cells with this approach, and then go back and discuss the biology behind what made this possible, as all previous protocols couldn't differentiate endocrine cells efficiently in planar culture. So we start out by expanding our human pluripotent stem cells, and initial cell density is one of the most critical parameters to have a successful differentiation. So we found that passaging the cells and seeding them at confluency and starting the differentiations the next day gives us the most consistent seeding density and the most consistent results in the actual differentiations. So then these stem cells are first driven out of pluripotency into definitive endoderm, shown here by copositivity of FOXA2 and SOX17. And then we specify the gut tube, and then with a high dose of retinoic acid, we turn on PDX1 to specify the pancreatic portion of this gut tube. And then the following stage is designed to turn on the transcription factor NKX61 in these pancreatic progenitors, which is critical for specifying the beta cells in the next stage, because if you don't, then the cells will resolve into alpha cells rather than beta cells. And so here you can see the dramatic changes in morphology as the cells transition from pancreatic progenitors at the beginning of stage 5 into endocrine cells in stage 6. And they often develop this golden hue under bright field microscope as they are becoming endocrine cells. And so at this point, most often we want to get the cells off the plate in order to do assays or transplant them into mice. So we can disperse them from the plate and put them on 6-well plates in an orbital shaker. And over the next week, they'll aggregate into these islet-like clusters that are about 200 to 300 microns in diameter, and they'll stay this size indefinitely in culture as long as you keep them on the shaker. So if we look at the composition of these clusters, we can see that most of these cells stain for C-peptide, which shows the beta cells, and then there are a few alpha cells and delta cells that stain for glucagon and somatostatin. And so, as I said before, in our system, by performing the differentiation in this planar format, and then only aggregating the clusters at the end, we found that our differentiations are actually better, and we've able to differentiate a much wider range of cell lines than we could with the Foley suspension protocol. And so here are beta cell clusters from 10 lines compared to primary human islets, including iPS cells from patients with various forms of diabetes, which opens the door to study different diabetic phenotypes in the lab. And we get anywhere between, so these clusters are about 100% endocrine cells, and about 20 to 60% of them are actually beta cells, in the, depending on the cell line. We can see that these cells secrete insulin in response to glucose stimulation, and the amount of insulin is dependent on the genetic background and a disease state of the stem cells, but our best performing lines secrete insulin on a level close to that of primary human islets. And as I mentioned, many of our lines generated with this hybrid protocol function better than cells made with the Foley suspension protocol. And so here you can see 2 iPS lines that now exhibit dynamic insulin secretion kinetics that did not when they were generated in our suspension flasks. And importantly, we can transplant these clusters under the kidney capsule of diabetic mice. And within a few weeks, cells generated with this hybrid version of the protocol in blue are able to normalize blood sugar levels. And they're able to basically cure the mice indefinitely unless we remove the graft, at which point they quickly revert back to being diabetic. The cells generated with the older suspension version of the protocol here in green also are able to cure the cells, but it takes a couple more weeks, showing that these are less mature than cells generated with our new version of the protocol. And so I'm going to kind of step back a little bit and discuss some of the biology behind what makes this possible, because if we try to do this method with the same factors used for the suspension protocol, we can make it all the way up to pancreatic progenitors, but we aren't able to get any beta cells out at the end. And so this is why all previous protocols had to be done in suspension at this point. So, what's different about culturing the cells on tissue culture plastic than in suspension? So if we look earlier at the pancreatic progenitor stage, and we take some clusters that we made in suspension, and we instead disperse them at this point and plate them down onto just tissue culture plastic coated with different ECM proteins, we can see that it inhibits the expression of this transcription factor called neurogenin-3, as well as some of its downstream targets. Whereas if we take those dispersed cells and we actually aggregate them back into clusters, expression actually goes up. And so why is this important? Well, all endocrine cells arise from neurogenin-3 positive cells. And so in the developing pancreatic ducts, some of these cells turn on neurogenin-3 and migrate away from the ducts and cluster to form your islets. So if neurogenin-3 turns on, it doesn't turn on, none of these cells migrate away and you don't get any endocrine cells. And so this effect was fairly independent of what extracellular matrix protein we used for coding, as long as the cells could strongly adhere to it. Some exceptions were some of the laminin isoforms where the cells didn't adhere well. They're coming off a plate. And so there we saw some upregulation of neurogenin-3. And so this inhibition of neurogenin 3 and hence endocrine induction appears to be a general effect of plating the cells rather than signaling from a particular extracellular matrix protein. And so the question becomes, what happens when you culture cells on tissue culture plastic? So plastic is a very stiff surface that resists the pulling forces of cells, which causes them to flatten out and polymerize their actin cytoskeleton, as you can see here by these stress fibers. Whereas if you plate them on something soft like a gel, or if they're in a cluster of cells, they'll remain spherical, and much of their actin remains depolymerized. And these differences in cytoskeletal state can influence different downstream signaling pathways. So kind of the model system for this is human mesenchymal stem cells, where it's known if you give them the same differentiation media and you plate them on something soft, they'll differentiate into fat cells, where if you differentiate them or plate them on something stiff, they'll differentiate into bone cells. So to kind of see if a similar effect was happening in our system, I screened a number of compounds that affected different parts of the cytoskeleton, and I got a hit with this compound called letrunculin A, which binds the monomer form of actin to prevent polymerization, which caused a huge upregulation of neurogenin 3. So it does appear that the state of the cytoskeletal polymerization within a cell is strongly influencing neurogenin 3 expression. And if we look at the ratio of actin in its monomer and polymerized form, we can see that the plated cells have the most polymerized actin, followed by the clusters and then reaggregated clusters, which matches up with the trend in neurogenin 3 expression that we see. And then adding letrunculin A, depolymerized actin even further, corresponding with our large increase in neurogenin 3 expression. And so this effect is very dose dependent. As you increase the letrunculin concentration, neurogenin 3 expression increases. And as you can see here in PDX1, pancreatic progenitors have very defined actin stress fibers, where these are eliminated with the addition of letrunculin A. And so neurogenin 3 is required for endocrine induction, but the timing of this expression is very important. So our stage 4 is designed to turn on the transcription factor NKX61, and these cells will ultimately become the beta cells. But if you add letrunculin during this stage, neurogenin 3 turns on, but it doesn't allow time for NKX61 to turn on. And so these cells will ultimately resolve into alpha cells rather than beta cells. Whereas our stage 5 is designed to direct cells toward an endocrine fate. So in the plated cells, neurogenin 3 doesn't turn on, we don't get any beta cells, so we don't get any insulin secretion out at the end. Or if we add it during stage 4, stage 6, we do get a little better induction of beta cells, whereas if we add it during stage 5, when we mean to induce endocrine, we get the best beta cell induction and cells that secrete the most insulin. So to look at this a bit further and kind of show the importance of the timing of these changes in cytoskeletal state and how they affect decision-making in pancreatic progenitors, we did single-cell RNA sequencing of cells treated with either letrunculin or nicotazole during stage 4. And so while letrunculin A depolymerizes the cytoskeleton, nicotazole depolymerizes microtubules. But one side effect of this is that it actually increases actin polymerization and tension within the cell, as you can see here. So we get a gradient of cytoskeletal states in these pancreatic progenitors to look at its effect on cell fate decisions. And so we can identify different pancreatic progenitor populations within these cells. First, we get our desired pancreatic population in blue here. We get high expression of PDX1 and NKX61. And then we have both an early and a late endocrine population in purple and orange. But because these endocrine cells were specified in stage 4 before NKX61 could turn on, most of these cells will become alpha cells rather than beta cells. And then we also actually have a population that expresses genes associated with the exocrine portion of the pancreas, such as PRSS1, which is trypsin, and this is a digestive enzyme that these exocrine cells secrete. So we can see that the ratios of these cell populations is drastically different depending on the condition. So the plated cells mostly consist of our desired pancreatic progenitor population, while majority of those letrunculin treated cells are this premature endocrine, as we saw before, while most of our nicotazole treated cells have actually shifted toward an exocrine lineage. So, the cytoskeletal actin polymerization, whether you have low, medium, or high, is strongly influencing fate decisions within these pancreatic progenitors. So just to kind of recap, being attached to the plate actually promotes a cytoskeletal state that helps turn on NKX6-1 in stage 4, but continuing to culture them like this traps them in this pancreatic progenitor state. And so once NKX6-1 turns on, we need to depolymerize the cytoskeleton to drive them out of this state into endocrine. And so the better controlling of the timing of these pancreatic transcription factors is most likely leading to the improved differentiation outcomes that we observe. So after a lot of optimization, we determined that a 1 micromolar treatment for the first 24 hours of this stage 5 was sufficient to initiate endocrine induction. And so if we look at chromogranin A, which marks the general endocrine population, we go from 20% endocrine up to 80 or more percent endocrine after this induction step. And so the cells that aren't treated with latrunculin are actually becoming other endodermal cell types, particularly liver, as pancreas and liver are very close developmentally. And so you can see here we have a lot of staining for alpha-fetoprotein, which is an early liver marker, as well as upregulation of genes associated with other endodermal lineages. And so this also has implications for other directed differentiations, endodermal differentiations as well. So for example, if we do actually an established protocol for making liver, adding nicotidol to increase actin polymerization actually increases the number of albumin positive cells. And as I mentioned before, nicotidol, by increasing actin polymerization, bias cells towards an exocrine phenotype during our beta cell differentiations. And so if we add it actually during a directed differentiation towards exocrine cells, it enhances the expression of trypsin, both in immunostating and mRNA, at the expense of a lot of our endocrine cells. So again, showing how cytoskeletal state is important for endodermal cell fate decisions. And so by knowing how your culture format influences cytoskeletal state and how this translates into differences in signaling with inside your cells, you could potentially improve your directed stem cell differentiations by modulating cytoskeletal state. So, your culture format is going to influence cytoskeletal state, and that can influence pathways within inside your cell. So finally, in addition to studying the cell biology of generating these cells and improving their performance, we're also interested in scaling this process up. So we have a JDRF-sponsored collaboration between an organization called the Advanced Regenerative Manufacturing Institute, or ARMI, to scale up and automate the manufacturing of beta cells, with the ultimate goal of providing a reliable cell source of research-grade cells that can be made available to investigators studying diabetes, as well as a demonstration of a proof of principle for larger beta cell production. And so in order to do this, we need to convert a lot of our small scale processes into equipment amenable for large scale production. So first we need larger culture vessels and settled on these Corning cell stacks. And fortunately, this process scales well with culture area, so we can do the differentiation in small well plates or larger flasks and get the same percentage of beta cells. And so here there didn't really seem to be a reason we couldn't go up further. So each of these cell stacks has a surface area of over 600 square centimeters. And in our manufacturing pipeline, we're now using one of these 5-layer stacks per production run. And so while not shown here, this 5-layer stack, it sits inside a custom manipulator that rotates the stack in order to drain media, and then rotates it back in order to pump more media in. So this is housed inside an incubator. And then every day of the differentiation, bags of media are clipped into the system, and then you basically push a button and it takes care of pumping out old media and pumping in new media. And so the next thing we had to do, during cell passaging and at the end during the aggregation step, we need to centrifuge the cells. And in order to have a closed system with minimal user handling, we settled on this Rotea centrifuge. And so this takes care of concentrating the cells, removing the dissociation reagent, and resuspending the cells back into a bag that we can clip back into the system. And so by doing this during cell passaging, we have no problems retaining our pluripotency markers. And so as I mentioned earlier, for the final aggregation step, we put a single cell suspension into 6-well plates on an orbital shaker to generate our islet-like clusters. And to translate this aggregation process into a large scale format, we're using these 3-liter reactors. And so kind of how this all works in practice is that the dispersed cells from the stack are pumped into a bag that are then attached to the rotea to remove the dissociation reagent, resuspend them, and it pumps them either into a new bag, or we can hook it directly up to the vertical wheel reactor for aggregation. So our yields for this differentiation process in a small format is typically between 500,000 to 1 million cells per square centimeter. And in a recent batch with the scaled up version, we're on the upper end of this range, generating over 3 billion cells. And so we can see expression of important markers such as insulin and chromogranin A and NKX61 are similar between batches made in a small format and those made in the 5-layer stack. And importantly, we can see that insulin secretion and glucose responsiveness is the same between cells differentiated with in a small scale format and those with the 5-layer stack and the automation of using the rotea for centrifuging and the aggregation in the 3-liter reactors. So the last step in this is being able to store and distribute the cells from these production runs. And so we're using a high capacity controlled rate freezer. And so these cells can be dispersed and frozen as single cells and then aggregated upon thaw. So you can see clusters here 7 days after thawing and aggregating into clusters, and we get these nice clusters that look like islets. Ideally, we would like to freeze them as clusters, but this is quite a bit harder. And so we're still working on this. So kind of our workflow now is that we can ship out frozen vials of the single cells and they can be aggregated on site, or we can aggregate them in-house and then ship the clusters live overnight. So in summary, I hope I've shown that culture format can influence cell biology during stem cell differentiations. And in our case, pancreatic progenitors are strongly influenced by cytoskeletal state, and by depolymerizing actin at the right time allows for beta cells to be generated in a planar format. And by differentiating the cells in planar culture and then only aggregating the cells out at the end, we're able to differentiate a much wider range of cell lines that are often more functional. And so this hybrid format has allowed us to easily scale up with minimal optimization of the actual differentiation protocol, so that we're now able to generate billions of cells per batch without losing any cell quality. So with that, I'd like to thank my lab at WashU here and the process development team at Army for helping us scale this process up. So I will take any questions.
Any questions?
So how many cells do you need to inject to each patient?
It depends how good the cells are. Like Vertex didn't release how many cells they're injecting, but I think it's on the order of billions, several billion. So like, you know, a run with our, you know, our 5-layer stack would hopefully be enough to cure, you know, a patient. But again, if we, you know, continue to make the cells better, you know, make them twice as good, you have to transplant half as many, which is quite significant in terms of, you know, a couple billion cells is a lot of volume to inject into a person. So yeah.
So kind of similar to my question to Dr. Lee, you mentioned the clinical trial is allergenic. I mean, when you know someone has diabetes, you obviously have a lot longer time to just make their own iPS cells differentiate.
Yeah.
Autogenic.
There is also the issue of that type 1 diabetes is an autoimmune disorder to begin with. So you could run into that issue if you do an autologous method. So, you know, to avoid the immune problem, you know, people for a while have been trying to do encapsulation devices. But there's also now efforts to make basically the cells hypoimmune. So you basically hide them from the immune system, which is I think is the method that's going to work first. So. And
the Vertex clinical trial is using the suspension culture method?
Yeah. So that's kind of based on their original protocol.
Yeah.
They don't publish or release any details of what they've done since that. So it's kind of hard to compare how well our cells are functioning now to theirs. But yeah, so like I said, the suspension protocol works, and for a few lines, it does work well. But if you're trying to do different lines, just the suspension culture just doesn't work for a lot of them. And so this, I don't know which one will eventually be used, clinically, but at least in a lab-based setting for, studying diabetes, this method works a lot better. We can do it in 6-well plates to do, miniaturize it a lot to do, lab scale experiments, whereas the suspension culture, you kind of have to do large volumes in order to actually have the suspension culture work.
So with the planar culture, before you aggregate the cells,
Mhm.
how functional are they?
They're functional on they're fully differentiated and ready to go on the plate. Whereas before we kind of found the bottleneck of, at the pancreatic progenitor stage, you couldn't make the cells on the plate. You had to aggregate them at some point. Nobody really knew why. So yeah.
But you showed the difference in transcription profile between the like before and after aggregation. So as far as your glucose test, response test, do you see a difference with or without aggregation?
So yeah, usually they function better after we actually aggregate them into the clusters. So they're fully differentiated on the plate, but we found that, yeah, their function improves if we actually aggregate them into clusters. Yeah.
When you do the aggregates, you compare the aggregates and you are making a single cells and injecting to the patients, right?
Sorry, say that again.
You are making aggregates and you are making single cells and giving to the patients.
So we make them as single cells, but then we disperse them and aggregate them. So everything that we always transplant or do any assays with are always those aggregates. Yeah.
No, but if you make the aggregates to single cell, and have you checked the insulin level?
Yeah, so we can disperse the clusters and plate them down to do assays with them, and they still function fine. But there's a weird thing where if we differentiate them on the plate, and if they don't function as well unless we disperse them and either aggregate them into clusters, or they do work better if we disperse them and plate them back down. So there's some kind of resetting of the cells when we disperse them. So yeah.
Hi, thank you for the wonderful presentation. I was wondering if de-differentiation is ever an issue that you've run into, specifically with the mature like islet cells. How stable are those?
They're very stable, so we can culture them for months in vitro, and they won't de-differentiate. They do lose their ability to be glucose responsive, and so then they secrete less insulin after about 3 months. But they will kind of retain their identity. It works if we transplant them into mice, the cells actually mature a lot more. So there's something of the in vivo environment that helps the cells mature, and they continue to, their insulin secretion actually improves in vivo over the long term, and can, we've had them in mice over a year and they're still cured. Whereas in vitro, they do eventually lose their function. And this is also known for primary human islets. Like if we get them, they kind of rapidly lose their ability to secrete insulin in vitro culture over a period of a couple of weeks. So.
No more questions? All right, thanks, Dr. Hogrebe again.